you. That's pretty good timing. I've never had an introduction occurring while a video has been playing of a beautiful sample being taken. I think most of you could see that sample, and we don't want to necessarily watch all the details of, there we go, the sample being aspirated by that remotely operated vehicle in the bottom of Yellowstone Lake. But here's a filamentous streamer community that we are studying, and there's a member here tonight, Dr. Luke McKay, that is working on characterizing the streamer habitat. This particular sample was just taken in 2016, but we also have some more from 2017 and hope to have a few more from this summer that are much like this. This particular sample is about 120 meters deep, just east of Stevenson Island in Yellowstone Lake. pH is probably 4.5 to 5, so it's not that extreme in terms of pH, but the temperatures down at the bottom of the lake can get quite high. The vent temperature of that vent fluid, of course, it's very hard to get an accurate measurement mixing with cold lake water, but as high as 140 centigrade or Celsius, that's probably about 240 Fahrenheit. We'll talk about the temperature scales on them in a little bit. I could not embed that particular video because that video is very large, and
Hi.
Pardon?
You asked for technical support or something?
Oh, no, I think we got it figured out. We're trying to figure out the way.
Yeah. So the next one.
Okay.
Yeah.
Yeah, thank you very much. So, exploring hotspots for thermophiles. This particular image is taken in Northern Dome, but it's in a community much like what you just saw in the video. So this is a scanning electron micrograph. You can see the scale bar of 1 micron there. And what we're looking at is elemental sulfur, these nice beautiful rhombohedral shapes with cells. And we're also looking at this network of nanowires that is draping over the whole community. And there's been a lot of work on whether or not these particular types of wires can conduct electrons through cytochrome proteins. So it's quite interesting, and I've imaged these types of nanowires in many, many examples. But, so you think about that sulfur community, it's an extremely hydrophobic environment and wants to coalesce on itself. And then you look at it close up like this, you also see that it's veiled with a network of nanowires, very hard to disperse this material. And that's why it survives like that down in the, hanging on for dear life in those high temperature vents that are quite active. Well, so back in about 98 or so, there were several people in the Land Resources Department and also Department of Plant Pathology. 4 of us got together and decided that it would be nice to start exploring Yellowstone in more detail. Mark Young, John Wraith, Tim McDermott, and myself started the thermal biology group. Dave Ward had already been working in Yellowstone for several decades, and he's a member of our faculty here as well. And so there had been a lot of interest in Yellowstone environments. But we got a big project from NASA, exobiology, to look at extant life forms in Yellowstone. And of course, there's good evidence that thermophilic or thermal conditions were likely very important in the origin of life. And NASA has a very big program trying to look at habitats, extreme habitats that might represent environments that could be found on other planets. I think they should broaden their view of what life is. They're at the same time, they're looking for extreme examples of life on Earth, and that's all we have to study. Yellowstone has at least at the moment, we're getting closer, but so clues for extraterrestrial life. NASA funds a lot of work, not only in hot places like Yellowstone, but also cold environments, dry environments. So we were lucky enough to get their attention, and we ended up bringing in a lot of funds for MSU as a result of that. And it lasted the funding from NASA lasted nearly 10 years. So it was a lot of good programmatic funding, something we're shy of at the moment. Programmatic money to actually invest and try and answer questions where you're not always having to turn around and worry about funding agencies every year or every other year or whatever. So this image is kind of fun because there's all three domains of life here. And this was in the middle of winter as well. This was March 2nd, probably it would have been 99 or 2000. And it was very cold. It was about -10, kind of like a little bit colder than it is tonight, but not too dissimilar. Anyway, you can see three domains of life here, and the spider is interested in flies that are laying eggs in the cyanidia, the phototrophic algae. It's actually a eukarya, like us, but cyanidia in green, and then you see this abrupt isotherm or temperature line there where you can go from the cyanidia to the elemental sulfur, and that's about 54 degrees centigrade right there. It's a great shot, and Yellowstone, it's an oasis. It's a geographic island where you can have tropical species growing in the middle of winter. There's a couple of plants that grow all year long. A little bit about Yellowstone. So, as you may know, there's been several big explosions at Yellowstone. The most recent about 630,000 years ago, then one at 1.2 or 1.3, and then another one about 2 million years ago. But you can track the hot spot all the way down through what is now southeastern Idaho and then southwestern Idaho. And the Yellowstone hotspot has been erupting for the last 22 million years, based on good geological evidence. And so the largest, or some of the bigger eruptions, though, have occurred in these three eruptions. And in fact, here's the size of St. Helens, and since we've lived through that, at least some of us have. Everybody remember St. Helens? 1980. Bruce knows. Fred? What was it, 81? Okay, so you can see the size of St. Helens here and then compare it to the Yellowstone eruptions. That's the volume of material that was moved out of the Yellowstone, some of the most recent Yellowstone eruptions. No comparison. And the ash fall, which has been mapped from those eruptions, looks something like this, and I'm not going to spend time with each of the different eruptions, but you can see that it's covering 2/3 of the United States. And so that's why people get excited about whether the supervolcano might erupt. I'm not here to tell you if it will occur soon, and we obviously have a lot of factors to talk about. But tonight's talk is not about that, so we'll leave that for Lisa Morgan to talk about, and other volcanologists who are current on those questions. Most life forms on Earth, 0 to 50 degrees Celsius optimum, and a reminder that conversion to Fahrenheit at 0 to 50 corresponds to about 32 to 122 Fahrenheit. And a couple of other reminders, at sea level, water boils at 100 centigrade or Celsius, 212 Fahrenheit. If I remember, I don't use the Fahrenheit scale that much anymore in some regards, not for science stuff, and so I tend to think in terms of centigrade. Interestingly, I was looking, I didn't know this, but Celsius is the name of the guy that actually invented the Celsius scale, and originally he had it from 0 to 100, but 0 was boiling. No kidding. And 100 was freezing. Very interesting. Well, that's a side note. At YNP elevation, 92 centigrade is boiling and 198 Fahrenheit. So the higher elevation, you get a little lower boiling temperature. The thermophile then is a type of extremophile that loves heat, of course. Organisms that are generally in this range would have optimum temperatures of 40 to 120 centigrade. And then we also have a class of organisms that live or thrive. It's not that they live and are having a hard time. No, this is their optimum temperature. You have to keep that in mind. Hyperthermophiles greater than 80 degrees. So there's a small window in Yellowstone where we would see hyperthermophiles really. I tend to bring this hyperthermophile limit down to 75 myself, but maybe it's just because I want to be a hyperthermophile. And there's not that many environments in Yellowstone that are above 80. There are plenty, but some of the ones that I was studying and some of the ones that are more interesting, at least the terrestrial ones, are more in the 75 range. Anyway, there's been a lot of interest in unique characteristics of thermostable proteins, enzymes, and possible engineering advantages. It's good to keep that in mind. I'm not going to focus a lot on commercialization of thermophiles because that's not what I do. But it's important to recognize that thermal stable enzymes can be highly stable for long time frames and resistant to different types of solvent, resist contamination from bacteria and other lower temperature organisms. The reaction specificity can be very, very high, and the rate of reactions can be very high. And of course, as you maybe know or are well familiar with DNA polymerases, which cause the molecular revolution, the fact that someone came along and realized that a high temperature DNA polymerase could, when added with the 4 bases necessary to form DNA, actually, and as long as you have a template, a strand of DNA that you denature in the reaction, you unwind it, and then get the 4 bases with an enzyme, a high temperature polymerase, you can make a lot of copies of DNA that then can be measured. And that, of course, that technology caused the molecular revolution, and so it's perhaps the most familiar thermal stable enzyme, these polymerase, and then that are used in the polymerase chain reaction. And so, like I was saying, the application of a segment of DNA across several orders of magnitude, you can generate a lot of particular DNA sequence for then subsequent measurement. And that's how we're doing genomes now, you know, to a degree. There's other technologies that have evolved since then as well. But amplification, we would not be where we are if it hadn't been for the application of high temperature polymerases. That came, TAC, the original one, TAC DNA polymerase came from Thermus aquaticus, originally isolated from Yellowstone. And now there's a lot of different types of polymerases, but they all come from thermophiles, or those are the ones that have been most applicable in the molecular biotechnology areas. You can see this one's from an organism called Pyrococcus furiosus. It's about 20 times more stable than even TAC polymerase. At 95 centigrade. So that's where the amplification is done at 95 centigrade, near the obviously close to the optimum for that enzyme, because that's what the organism was thriving at. It's made that company a lot of money. It's not the only thermal stable enzymes. You don't hear a lot of. They're a big market for thermal stable amylases, and if you're a home brewer, check this out, firm fast alpha amylase enzyme, and sufficient for mashing up to 20 pounds of starch. You can get your potatoes or corn or whatever you want ready here with before ethanolic fermentation here with thermal stable amylases. And there's a big industrial a lot of industrial interest in thermal stable amylases, chitinases, cellulases, lipases, a lot of those used in the food industry and degrading plant material. So just one word on benefit sharing in the park, because I think it's an important subject, of course, and I won't talk much about it, but just want to bring it up here that because of Thermus aquaticus, the development of PCR resulted in this large business. Federal legislation authorizes the park now to negotiate benefit sharing agreements, and they can, NPS, the National Park Service, can receive monetary and non-monetary benefits from discoveries or inventions from research originating with a permit. There's not a lot of examples of these now, now that the money's been made, but you never know. Some of these could be important in the future. There's, right now, I do not, on the microbial side, there may only be 1 to 3 benefit sharing agreements in Yellowstone right now. It's not oversubscribed to at the moment. The first one was with a company named Diversa, who is now out of business, and they, in return, their benefit to the park, instead of paying the park, they sequenced the original wolves that were introduced. And at the time, it was a lot of money, because that was before you could do a wolf for $100 or whatever. I don't know. I'm not sure what it is now. It's cheap. Anybody here have had their own genome sequenced? It's coming, you know. It's cheap now. Work presented in the current talk is performed under these permit numbers, 5568, 5068, and there's no commercial goals in our work, and so directly, and so I've never entered into any benefit sharing plan. Our goals are to understand the distribution of different thermophiles in Yellowstone, and I've been really big on my original background, as Bruce alluded to, in environmental chemistry. I'm really a geochemist, interested in how geochemical variables influence the distribution of life, and these microbes are utilizing chemicals, so it's a very good fit. And we're very interested in the role of variables like temperature, pH, sulfide, and oxygen, and also what are the various functional properties of organisms that are living in these environments. You know, what, in other words, what allows the organisms to live there? What proteins, what processes, what pathways, etc. Some things we take for granted, perhaps, and this is a nice reminder of a temperature scale, just so we get dialed in again to, of course, there are organisms that live all the way down, even below zero, but you can see on this scale that, notice they have sulfur-dependent archaea and methane-dependent, or methane-producing archaea up here in the highest possible ranges. Of course, you know, some of these organisms do live at lower temperatures also, but it gives you a sense of the distribution of different types of life forms as a function of temperature in the centigrade Celsius scale. The other parameter that becomes really important is pH. And so on this plot, what we're looking at are the number of different geothermal features in Yellowstone on the y-axis over here. And you can see it's only for 7,700 geothermal features, and there's probably about 14,000 in the park that are currently mapped. So this isn't all of the geothermal features, but a good 55% of them. And so it gives you a sense of the trends of the pH range. So you can see a bimodal distribution with a pH of about 2.5 on this lower one, and then pH of about 6.5 on this other, let's say, group. But there's all kinds of hybrids in between. As a reminder of the pH scale, and I think this becomes an important parameter and variable that we throw around, and so pH is the log, the minus log actually, of hydrogen ion concentration. So you can see the pH is a log scale, goes from 0 to 14, and battery acids down here are nice and low, pH 0 all the way up to pH 14, where you get soaps and detergents. And you can see here's an example in the upper one of a pH 3 system, of kind of a boiling mud pot. People would call this it falls into the realm of solfatara-like, and a lot of sulfur oxidation, and that often is one of the reasons why it's a lower pH. You can see these pHs are very common in 2 to 3. We also have a lot of systems like Octopus Spring here on the bottom, where the pH is about 8, which is just perfect for a lot of types of organisms, and even more bacteria that will thrive. And you can see phototrophs here lining the octopus channel. In fact, I'll say a word about phototrophs here. Isn't that a beautiful image? Miles O'Brien from CNN took that picture. I want to give him credit right now because it's one of the coolest darn photos that I have of Yellowstone, and it's an outflow channel with phototrophs growing. One of the things that you might notice in Yellowstone is color, and a lot of the geothermal channels, a lot of different colors. And of course, it's great if you can start thinking about how to convert colors then to types of organisms and what that must mean about the conditions of the spring. And probably what the likely temperatures are. Once you start to pick up the patterns of what is possible down there. So for example, in this shot, where do you think the brown is from and the red? Beautiful. Beautiful answer, but it's not the right answer. I don't see Ed Dratz here, but Ed Dratz called me up on the phone one time and said, Bill, what's all this red color doing down at Mammoth? He said, that must be iron coming out, you know? And it's like these reds and browns, it's photosynthetic pigment of phototrophs. And the iron levels in these waters here about pH 8 are extremely low, and you're not going to see much iron precipitation in this particular circumstance. But you do see a lot of photosynthetic pigments that, you know, they're not green like we're accustomed to seeing. That's only one type of phototroph that absorbs light at a certain wavelength. And of course you have other organisms that are absorbing light at different wavelengths, and hence being visible as different colors. Once you start picking up on this pattern, it becomes extremely helpful. I know from this shot, for example, that there's probably no sulfide in this water because it's supporting of a lot of cyanobacteria. And I'm going to guess that the pH is about 8, 6 to 7 maybe, coming out of the ground, and then maybe increasing down the channel, supporting these types of phototrophs. So once you pick up the pattern, it can become quite useful. Now I want to contrast phototrophy over on one side. That's what we were just talking about there. I'm not going to talk much more about phototrophy, and that's why I'm trying to kill it now so that I can get on with the more interesting stuff of chemotrophs, because I'm a geochemist. And we're going to talk about chemicals serving just like the sun. Chemicals are driving life. It could be completely dark and these organisms wouldn't care, okay? So if there is any diel phenomena, it's only through the indirect reactions of other things that could be changing with light, like free radicals, peroxide, and things like that. And those could have very real effects on cell growth, etc. But what we're talking about here are chemicals that drive energy and metabolism. And so these are called chemotrophs. You can see there's organisms that live on organic chemicals like us, chemoorganotrophs, fancy words, nothing too big about them really when it comes right down to it. Chemolithotroph, litho being rock. So these are organisms that are going to be getting their chemical energy from hydrogen, H2S, ferrous iron, ammonium, etc. What we mean by etc. here is other reduced ion compounds that have extra electrons that organisms could utilize for an energy source. So we went on in search for chemotrophs. And I really enjoyed the field in Yellowstone. It was probably one of the best things that ever happened to me, really. And we were able to visit all kinds of different springs, and this just gives you a sense of some of our adventures in Broad Creek, which is 25 miles north of Yellowstone Lake, packing in a lot of scientific gear so that we could do chemical analysis in the field. A lot of chemicals you can't store, or parameters you might want to measure, like arsenate versus arsenite. Two different forms of arsenic. You need to measure that in the field, otherwise it won't survive storage. Or iron 2 and iron 3, you got to measure that in the field. Things like pH, okay, you really need to measure that in the field. So and we were doing gas analysis in the field with this portable gas chromatograph when it worked. It was sometimes a problem. So the main point of the last slide, and before it leaves my mind anyway, is to kind of give you a sense that, okay, we made a lot of measurements, geochemical measurements. We also took a lot of samples for biomass analysis through molecular work, trying to get the DNA signatures and then look at the genomes of these organisms. So that's the combination of approaches that we were using, and a lot of microscopy, because I do a lot of scanning electron microscopy, which you'll see some examples coming up here. So here's Norris Geyser Basin. I'm going to get into some of the example types of sites that we work with, and this is acid sulfate chloride spring in Norris Geyser Basin, and you can see here they call this Dragon Spring. It's got a pH of 3 here at the source. You have water coming out, and you see all this elemental sulfur depositing, and you also see a band of HFO, which really meaning to hydrospheric oxide. Just think of it as iron oxide rust forming from the reduced iron that's in the source waters. So if you keep your eyes on the sulfur deposition zone, what you see here is a lot of sulfur spheres, and then the growth of sulfur rhombs, the rhombohedral shaped sulfur. And a lot of this is abiotic, but can also be mediated by some microorganisms that utilize sulfide and convert it to sulfur. But here is a bunch of elemental sulfur crystals just stacked up perfectly in these environments. Just beautiful. And you can see one sphere there. It's like a game of hide and seek. This is probably abiotic precipitated sulfur. But organisms are there, and if we go downstream a little bit for a minute to the iron oxide zones here, these rust-colored zones, I want to show you a close-up there, where we get the biomineralization of high arsenic iron oxides. You can see the sheaths that develop here around a cell, and you can see the cell, where the cell was in that little hole there. That's where the filament was growing. And then iron oxides have been biomineralized on the outside. And we know the chemical composition because we can do some fancy spectroscopy and analyze iron and arsenic. Turns out that most of these sheaths are extremely high on arsenic as well, that form in these acid conditions. It wouldn't be forming if it wasn't for these organisms, though. And interestingly, the filament that's getting biomineralized here is not the organism that's oxidizing the iron. It's sort of it's what it gets for growing in this environment. One of my favorite sites, and I'm going to stay with iron oxides for a minute because we learned a lot about iron oxides, and I want to spend a little time on those. Here we are at a Kindness Geyser. You can see this location if you're on the boardwalk, the back basin, back loop in Norris Geyser Basin. And beautiful terraces, probably the best in the whole park as far as iron oxide terraces. You look at how level that is. It's like a microbial beaver dam. It really is a series of thin film bioreactors, and it only lasts for a couple meters. It's kind of interesting. I'll show you some video of that here, and we'll get a sense in a minute. This, if you look at these iron oxides in close up, you can see here's an enrichment where we're able to grow several different types of organisms from this environment. And if you enrich for the organisms, you can see them everywhere. This is the Nicodins prep. which is a method of enriching the cells. And you have to look at it, but you can see the filaments in here and the cocci-shaped organisms. They tend to become a little deflated with the scanning electron microscopy. The holes is filter paper, that's 0.2 microns. So if you're wondering what those holes are, in both of those electron microscope images, it's the filter that's being used to filter the cells out. So have a look at, oops. There we go. What you have in these types of systems of echinus there is just a really good example of a very, very thin film of water. Comes out of the ground as primarily ferrous iron, and so there's very little oxygen in the source spring, but as it comes over those terraces, that's a great way to oxygenate the water because we got 20% oxygen on top of it. And the organisms that are oxidizing the iron need that oxygen as an electron acceptor, so it's a perfect combination. And you wouldn't get these nice, beautiful terraces like that without the microbes oxidizing that iron too. It's probably, literally, I think it's the prettiest iron oxide environment in the park, echinus. You can see some other features here that some other cells in echinus, but I also wanted to just give you a sense of the visual range in types of iron oxides that form in these types of circumstances. Very similar circumstances, but different iron oxides, interestingly, depending on the chemistry. We go from kind of a ferrihydrite here in echinus all the way to hematite in epigeosis coat with this real interesting red, almost rust or barn red color, all the way to a site at Rainbow Spring where there's a lot of jarosite, which is a mineral, iron potassium sulfate mineral. So the local conditions can be a little different, but what we found is some of the main organisms that are oxidizing iron are the same across those three systems, even though the mineralogy is a little different. But beautiful range of types of iron oxides. We started getting interested in how these systems form, and so one of our students, Jake Beam, and we all used to work together in the field, but Jake had done this project where we looked at slides and inserted into these springs for different lengths of time. Here you see some 7-day-old slides and some 15-day-old slides. Notice at 7 days, about the only thing you see are these organisms we call hydrogenobaculum, which are very early colonizers. In fact, they come in within a day. They start to grow on the slides very quickly. So you know that they're turning over and constantly growing on the surface of these iron oxide mats at a very rapid rate. Within 2 weeks, you can see probably the first archaea actually imaged in situ. I mean, in a real environment. Not many people take pictures of archaea in where they live. That's what I'm saying. And here you can see here's some microcolonies circled with in the white dashed lines, and a close-up of one of those microcolonies showing, and don't really know just from this kind of an experiment what organism that is. We can surmise what it is, though, with our companion molecular measurements. And it's probably the iron oxidizer metallospira, which we isolated. Now, within 15 days, we also see something very interesting, that viruses start to infect those archaea very rapidly. You can see what we call bottle-shaped viruses, and they're on the neighborhood of 100 nanometers long. But you can see them. They've just attacked this one cell in the middle and blown it open, and it's become a viro cell. That's a cell that turns into a virus factory. So look at the close-up here of the bottle-shaped viruses either coming in or out of an archaeal cell. And then over here, also within 2 weeks, you see these filamentous viruses. They look like little miniature small nematodes. So there's 2 virus types just in this one image. And of course, as Mark Young would remind us, viruses are probably controlling the larger organisms, the archaea and bacteria, in terms of their turnover rates, and become very important. It's a completely unexplored world out there in terms of viruses, very unexplored. Mark has had a program here in virology working on viruses in the Sulfur Systems. And he's only worked with a couple of types because it takes a lot of effort, and he's done some very good mechanistic work. The point is that there are so many novel viruses that have not been described, like the ones that I just showed you. We don't know for sure who those are, even though we have a lot of sequence data that corresponds to viruses. But anyway, take a look at these ridges that form. After 30 days, you can start to see the iron oxides forming these micro terracets, we call them. And you can see sometimes they're 20 microns apart. Notice that what they are is a bunch of cells. The ridges are actually cells growing up into the water, using the oxygen and oxidizing the iron. And here's a good example of what those terracets turn into. One of the things that we don't understand at all is how the terracets, I think we have, what we learned is how they start, but how they actually coalesce into the larger terraces, and what controls that dynamic is, I guess we could probably surmise it's a lot of feedback with the hydrodynamics and the water, but how the small little terracets form or turn into the big ones, that's something that we haven't studied at all. Now, I want to emphasize some of the back to the filamentous streamer communities in Yellowstone. There's really 3 major lineages. Ones that, like low pH systems over here on your left, the sulfur and the iron examples, those are pH 3. And then we have more neutral systems in the middle, but they have very high sulfide. And we have systems over here on the right, which higher pH, but no sulfide. And all of these are the same basic organism type, although they support other community members. But it's the same basic organism. You can see it here in this beautiful example of sulfur streamers at low pH in Norris Geyser Basin.
Yeah, 62. I have 62.1, but otherwise it's 62.
Beautiful systems. And they're oxidizing sulfur. Here's what they look like. Filaments with elemental sulfur ROMs, and these organisms are likely capable of oxidizing either the H2S sulfide or the elemental sulfur and oxidizing that to sulfate. If you look in the same pH systems, just a little down gradient, you get these kinds of what we call gray streamers, but it's the same organism. That's a short video, but play it again. You can see those streamers responding to the gas coming out of that bubbler. It's either likely they're responding potentially to either hydrogen gas and or H2S. And they have capabilities to oxidize hydrogen as well. That organism type, what we figured out with the genome sequencing, basically that gray streamer is a perfect mix of hydrogenobaculum and metallosphaera. Hydrogenobaculum is the filament you see here, and you can even see some cellular material coming out of the sheath there, sticking its tongue out at us. That's hydrogenobaculum. Metallosphaera is a coccoid archaea that you can't see here very well. It could be some of these things on the surface, but metallosphaera doesn't form a sheath, so it doesn't accumulate iron oxides. It's kind of an interesting relationship. But metallosphaera is the one that's oxidizing the iron. Now, a system that we've all probably walked around, Mammoth Hot Springs, this is narrow gauge, beautiful site. What's the red color? Now we've seen both examples. We've seen iron and we've seen red pigment. So it's one of those two. Bruce has been burned once already, so it's pigment this time. We're back to pigment at Mammoth. And these are phototrophs that you see growing at lower temperatures. When I say lower temperatures, temperatures below about 70 degrees centigrade. That's the upper limit for phototrophs. And Mammoth, of course, you get some beautiful terraces at Mammoth. These are all carbonates, which is kind of a coincidental situation at Mammoth, where the hot water is coming through the Madison limestones, dissolving those carbonates at depth and reprecipitating them at the surface. And that's really what these terraces are formed out of. And the organisms could care less, other than the fact that they're getting entombed with carbonates at a very rapid rate. And that's probably what they care most about. You can see the kind of lips that form up near the source waters of Mammoth Hot Springs. And you can, I think, through the steam, get an idea of some of the filamentous behavior of the sulfury hydrogenidium organism, they call it, that grows in these systems. Here's what it looks like. It's a filamentous organism again. It's an aquacales, very similar to the ones that are growing in the sulfur streamers. And here's a picture of the calcium carbonate, the aragonite needles that are forming. And these are, it's an abiotic process, except for the fact that the biota might nucleate, or maybe a place where the template, where the precipitation can occur. But otherwise, these organisms are utilizing sulfur in these systems. Okay, so we jump over to calcite springs. Kind of a misnomer. There's no calcite there. It's kind of a funny circumstance. Here we were just at Mammoth, where there is some calcite down gradient, but it's mainly aragonite that's forming up at high temperature. Anyway, it's calcium carbonate. At least they got that right. Over at calcite springs, there's not a lick of calcium or calcite in this system. What it is that's weird. I thought I had another shot in there somewhere, but I don't know what happened to it. But you can see the black solid phases in there are the pyrite that's forming, iron sulfides. So this is a really sulfidic system, and the yellow streamers that you see in the solid phases, that's elemental sulfur. So these organisms too are utilizing sulfides. Now, you wouldn't expect the lake necessarily to be a great place to study thermophiles, but as it turns out, there's some wonderful vents at the bottom of the lake. And this work preceded us. Really, there had been people working in vents in the bottom of Yellowstone Lake for some time. And we got lucky enough to be part of a project in 07 and 08 that had to do with some of these vents. And then recently, got involved in a project. So this is kind of a hybrid of some material that took place, or work that took place in 07, 08, and some work that has just taken place in the last year. So One of the ideas in a large lake system or even an ocean system is that hydrothermal vents might be ecosystem drivers, because you have reduced fluids providing organisms and bacteria that are living here. And maybe you saw those worms in that first video that were close to that thermal vent. You know, a lot of times you can have a increased eukaryotic activity near these warm vents because of the primary productivity that's occurring from the chemicals coming from the vents. This is not uncommon in hydrothermal vents in the ocean as well. So it's one reason to study these vents. Here you see the old boat, Luke. That's the old boat in the old ROV. We have a new one now that, well, we have it, but the guy that contracts this work, Dave Lavalvo, has a brand new boat and a brand new ROV, and that's what we're doing now in Stevenson Island at the deep vents, 130 meters. Temperature's extremely hot, 140 to 170. Whether or not there's organisms that are actually surviving those temperatures is another question. But you can see some bathymetry elevation work here, this very deep hole off the east side of Stevenson Island. And then, of course, in the older project, while we were filtering samples, we had a lot of help here in the parking lot of the Lake Dorm. We had to navigate by these bison every day, and let me tell you, it's a little bit scary. The entrance to the dorm is right here by those jugs. Fortunately, the bison, you know how they are. As long as you don't try and put your kid on them. So here's a couple of vents in the bottom of the lake. I think you can see some streamer material near the hot water there. And one more, it's not a long one. So these are vents that are variable depths, but the first one was about 30 meters and this one's about 50 meters. So they're not as deep as the Stevenson Island vents, but you can kind of imagine them. Well, these might not be that different than the systems on the terrestrial, the streamers that we see. And indeed, some of the same organisms are there, but interestingly, the concentrations of certain constituents like sulfide and hydrogen and methane are so much higher at the bottom of the lake that we've picked up some organisms that are responding to those chemicals where we've never seen those types of organisms in the terrestrial systems, especially methane and hydrogen, actually. This gives you a sense of, well, just pulling up the remotely operated vehicle from sampling and taking those pictures. And here's one of the yellow streamer samples that we pulled up from what we call inflated plain. You can see the distribution of organisms. I'm not going to go into great detail here on that, but you can see it's largely this Aquacales. That's the same thing that's central to the streamer communities and the terrestrial on the land systems. And so quite interesting. If you take some closer pictures, that's where the cover shot, we started with this one on the lower right. But you can see cells very clearly, even the coxoid type cells in these upper images, and then the sausage-shaped cells with the rhombohedral sulfur in the lower images, along with the web of nanowire material. Then we finally get to some systems that are, well, you might take, for example, and I call these low oxygen elemental sulfur systems, and the guy in the background here is actually in the audience. He can recognize himself, but nobody else can. He knows this shot. Zach J. Spent a lot of time with me in the field, probably more than anybody. And in any case, Zach studied these low sulfur systems, or high sulfur, low oxygen systems. And one of the things that's funny is, for the geochemistry, it's not all that different than some of the other kinds of springs. But one of the things that's very different is the hydrodynamic circumstances. Like Cistern Spring, it's largely a pool. We're working with an outflow channel, and so that there hasn't been a lot of significant oxygenation yet. You can form iron oxides from cistern probably, and Monarch, and some of these other types of pools but the water is not being mixed with oxygen at a rate quick enough to get those kinds of organisms. So we're sort of stuck with the sulfur cycle, if you will. And we get oxidation of sulfide to elemental sulfur, and you can see the yellow sulfur all around the perimeter of the pool. But it started to teach me that there was a lot of hydrodynamic or just hydrologic feedback on what then pushing the geochemistry, which then pushed the organisms to either be there or not, depending on their metabolisms. And in fact, this is perhaps a complicated diagram. What it is, a little bit of context is simply that a lot of people are interested in correlating where microbes are with geochemistry. And so if you go out and measure everything and throw it to a statistical model and act like you don't know anything, you don't get very satisfying results. Got to use your head. I started thinking about this, at least my head took me in the direction of, everything's a rule-based system. It's like a decision tree format. It's not about R-squareds and correlations with a certain parameter. You either have it or you don't have it. And for example, the phototrophs can't live above 74 centigrade Celsius because the photosynthetic apparatus breaks down. Biochemically, you are not going to have phototrophs above 74. So it's a decision tree. It's like, look, the biochemistry doesn't support it. There's no reason to go out and try and take a bunch of samples at 80 degrees and correlate temperature with phototrophs. They're not going to be there. So I started to think about what are the minimum number of variables needed to really identify what the primary organism types are. And what I came up with was pH, temperature. You can see in the brown boxes here, 1, 2, 3, sulfide, dissolved sulfur or sulfide. And that kind of serves as a surrogate variable for oxygen. Then you need a little bit of hydrodynamic knowledge, and that's the fourth one. I'm not going to go into that one because it's more complicated. But just by knowing pH, temperature, sulfide, and then some hydrodynamic positional things, like whether you're in a flow regime or a pool. If you're in a flow regime, then you're going to get oxygen in faster. And those variables could explain pretty much the types of communities that we were seeing, or that we see. You can see the green and phototrophs here, all up here because temperature is so important in establishing where phototrophy exists. The blue, the Oxide streamers, they exist in three different types of situations based on the three lineages that I showed you. Iron oxides form in only one kind of circumstance. And of course, there's plenty of things that we didn't study either on this sort of a decision tree type of format. If I could leave you with perhaps something a little heady here, but it's very important in terms of thinking about oxygen cycle. And this is what's driving a lot of the distribution of phototrophs. After we establish temperature and pH, then the issue is sulfide oxygen. That becomes extremely important. So we think about source water coming out of the ground with reduced fluids and oxygen streaming in, and we're having dissolved gases like hydrogen, methane, CO2, and H2S. These gases are actually leaving the system as you go down gradient. And of course, this air-water gas exchange is one example of the reaction we're interested in. How simple it is O2 gas equilibrating with O2 aqueous. You can see that's an extremely simple reaction. It's very complicated when it plays out in nature or in a lab because it's dependent on things like velocity, turbulence, air-water surface area. So all of those factors become really critical in establishing how much oxygen is getting into the system and what types of thermophiles are going to be there. that became a real eye-opener, and one that I'm still intrigued with. And that's why I'm talking about it, I guess, is because I'm still quite fascinated with the whole issue of oxygen. And it's interesting, it's quite important in terms of Earth's evolutionary history. The evolution of cytochrome oxidase, which is something that I've studied a lot in these systems. It's an enzyme older than presumably atmospheric oxygen. It's a catchy title because some people think that oxygen wasn't important until phototrophs came along and started producing it. But that history is, that story is going to slowly get put in its place. There's an early history of atmospheric oxygen, and there's a lot of work now that shows that life could have flourished on Earth even greater than 3.2 billion years ago on oxygen levels that were rising far earlier than we likely have thought, especially low levels of oxygen, which could have supported life, not the same levels of oxygen that we have now to support a lot of eukarya. So, in summary, if you look at the Earth microbiome, it's comprised of a lot of diverse organisms, and a lot of evidence that would suggest that the genetic machinery has been inherited from thermophiles. Thermophiles likely came first. Of course, there are people who believe that life originated in cold environments, and there is that argument in the literature. But I'll stick with the isotopic record and the phylogenetic record, which suggests that thermophiles are very, very near the base of the tree of life. And if you trust the tree of life, those near the base then are closer to the origin. Functional proteins involved in energy capture, generally what we found is they track very nicely with geochemical conditions. But it's not always a simple correlation. It's not like, it's more along the lines of yes or no. The organism has the capability to oxidize sulfur or hydrogen or arsenic, or it doesn't. And it's a decision tree kind of format. Oxygen is a very important electron acceptor for many thermophiles, not something that people thought. In fact, if you read the literature 20 years ago about archaea, it'll say that they're all anaerobes. None of them like oxygen. Nothing could be further from the truth, actually, when we look at the archaea now. There's a lot of archaea that love oxygen, maybe low levels of oxygen, like were around before phototrophs started producing a lot of oxygen. Very likely. So then because so much is driven by gas exchange and sulfide, or oxygen and sulfide, you have to realize that gas exchange and hydrodynamics, oxygen diffusion contribute as determinants where these organisms are distributed and located in different habitats. And remember, hot springs are unsafe to enter. We'll always watch the signs down there, and you'll see the funny signs. That's what I really need for part of this slide, is the funny sign that has the person scalding as a result of falling in the hot spring. It's not so funny. People have died. And along with this bison, probably was overwintering at Washburn Hot Springs. A lot of animals do this. They'll overwinter in hot spring environments, and because the ground's warm, and the thermal conditions have caused open forage for a while till they run out. And that's my guess. We've seen other dead bison up at Washburn as well. So took about, in fact, this picture was literally taken in '07, and we've been back there as recently as this fall. About 2 years ago, though, I think we could still see the bison head. Took a long time for this thing to completely degrade. But anyway, any questions? Yeah, Bruce.
So Bill, I'm trying to get the...
I'll repeat the question too.
Okay, I'm trying to get some understanding of what, when you talk about functionality, when you're talking about the geochemistry that's going on, and where most of the thinking was, and then suddenly, or gradually we started to understand the fact that there was a biochemistry going on as well. And it seems like there was feedbacks on those too. And that really complicates what those kinds of maybe outcomes are. And yet the distributions, you say, are fairly predictable based on, but it wasn't clear to me whether those are based solely on the geochemistry, which it sounds like it is, but there's something going on with biochemistry too. So I'm just trying to sort out, is that distribution part of which organisms are where? How much of that is biochemistry, how much of it is geochemistry?
Okay, so Bruce is wondering what are the contributions of geochem versus biochem in establishing the distribution, and perhaps I made it sound like it's all geochem. No, I'm just trying to lay out the connections are real. They're for real. It's not fake news kind of stuff. It's absolute. And it's directly, intimately associated with the biochemical feedback. I mean, an organism is only going to be in a certain spot because of this mix in geochem and the biochemical attributes of the organism. And that's what makes it so powerful. Because you can't, the organisms, they're going to have a range of habitats where they can survive. And we don't fully appreciate that, of course, in very many environments at all. Do we really appreciate the extent of gradients and the activity of organisms across a really small gradient? But nevertheless, what's absolute is, an organism's got a certain type of heme copper oxidase that can only grab oxygen at a certain level, a certain concentration. That's going to define the environment in which that organism can live. And that's why those streamers like to be in current, because that helps the turbulence and most likely, and oxygen, they can actually meet their oxygen demand based on, they set up shop where they can get that oxygen. So it's an intimate relationship between both. And I think that is, in my mind, what does make it so powerful, is because the geochem is telling you what the metabolism is. It's just that we need to learn that, and we're still learning. The geochem is in this range, and the physics are in this range, and then we're likely going to have these types of metabolic properties going on, which defines then, of course, who is it? What is it? Is it hydrogenobaculum or metallosphaera or some other organism type? Mm-hmm. Hopefully that helps. Yeah, Lou.
Do you know what causes the bimodal distribution of pH? Like of all the hot or all the features, there's the lower and the slightly higher pH. What is there two different types of rock underneath or something like that that's causing it?
Interestingly, it's not all just rock parent material, although the majority of parent material in Yellowstone is rhyolites. But because that's the volcanic rock source. When you get over the east side of the park, it's more andesitic, and the rock type changes. But it's very peculiar in terms of the kinds of formations that the water goes through. What explains the lower pH is a lot of old solfataras. So because it's an active volcano, there's always been a lot of SO2 and a lot of sulfur, very high sulfur. So there's deep solfataras, and sulfur oxidation creates hydrogen. So when you get extra hydrogen and the pH goes down, that's what's causing a lot of systems to be down in that pH 2-3 range. Meanwhile, you have other systems where it's not going through solfataras and going through rhyolite, which would be more pH neutral, potentially, or you're going through, like in the example with Mammoth, carbonates, and at the high CO2, that buffers that system at pH 6.8. And so that's one example of why you would get springs in this other range of 6 to 7-ish. One reason why the pH goes up is a lot of these systems are high CO2, and when the CO2 degasses, as it comes out of the ground, then the pH will tend to rise, or does rise. So a lot of those systems that come out of the ground at pH 6.5 will, if you go downstream a little bit, you'll measure a pH of 8. The pH will have gone up. But that helps explain what's separating that distribution. A lot of sulfur influence and sulfur oxidation on the low pH side, and then probably a more catch-all of types of conditions forming the higher pH stuff.
All right, well,
Yeah, Dr. Teets, one more question for Dr. Teets.
Is there a particular structural program which an organism decides to look for, as we see in some of the biological systems? I notice you have a very consistent pattern of adhesion between the organism or a portion of the organism, the chemical, and the substrate under which it operates, or against which it operates. Are they looking for a nutritional spot? Are they attempting to get a structural hold? Do they have any feeling in terms of how?
It's a great question. All these streamers, and Bill is asking about the adhesion. What causes these things to set down on a particular substrate and then hold on? You can see in some of these high current streams with the streamers, they're tied down, they're anchored. And a lot of times the streamer actually forms a little different structure there, but nobody's studied it. I haven't made much progress on what are the mechanisms of tie down. Really don't have a great answer for you on that. Gil Geyser was working on thermophilic or thermal adaptive adhesive compounds that cells produce to stick on the surfaces. And there's people that work on that. It's just I'm not that familiar with it, Bill. In these particular types of organisms, though, the Oxide, which form these streamer communities, there has been no work done on the mechanism of attachment. But yet, you can crank up the flow rates on these streams, and you cannot get them. You have to rip them off there. They're attached, and hence their ability to withstand a lot of hydrodynamic influence. But good question, and sorry, not real detailed in the response. But.
okay.